Hello, everyone. My name is Wafaa Rashed, the chair of Pan-African BGS Education and Research Initiative. I will give you lecture number 2, Understanding Polygenic Traits in the Basic Level Introduction to Polygenic Scores course. The main objectives of this lecture to define and explain polygenic traits, differentiate between monogenic and polygenic traits, provide examples of polygenic traits from literature, discuss the genetic and environmental factors influencing polygenic traits. And I will highlight the significance of polygenic traits in research and healthcare, and I will give you case studies. The main points that we will discuss, first, define polygenic traits and differentiate between monogenic traits and polygenic traits. Then provide examples about polygenic traits and discuss with you the genetic and environmental factors that affect polygenic traits. In addition to, what are challenges when you want to study polygenic traits? The main challenges. Research techniques used for studying polygenic traits. Ethical considerations, which are very important when you are doing polygenic trait research. The significance of polygenic traits in research and healthcare. Case study about polygenic traits in personalized medicine. And finally, applications in African context. Let's start with the definition of polygenic traits. Polygenic traits are traits controlled by multiple genes. From the term polygenes means multiple genes rather than a single gene. So each gene's influence is typically small but additive. So each one contributing, no, it's aggregation of these small effects finally affects the overall phenotype. Again, each gene's influence is typically small but additive, contributing to the overall phenotype. And the additive is very important characteristic of the polygenic trait, and we will discuss in the following slides. Examples of this for height involve hundreds of genes, each contributing a small amount to the final height, the final polygenic trait. If we consider height as a polygenic trait, one of the polygenic traits. Skin color, again, determined by multiple genes controlling melanin production. Or weight, influenced by genes regulating metabolism, appetite, and fat storage. Intelligence, a complex trait influenced by numerous genetic and environmental factors. Blood pressure, polygenic and influenced by genes affecting vascular function and cell balance, and many others. Susceptibility to diseases, conditions like diabetes and heart disease have polygenic components. So, what is the difference between monogenic and polygenic traits? For monogenic traits, they are traits controlled by, from the term mono, single gene. Examples for this, cystic fibrosis. Cystic fibrosis is caused by errors or mutations in the cystic fibrosis transmembrane conductance regulator, or CFTR gene, which results in either no CFTR protein being made or a malformed CFTR protein that cannot perform its key function in the cell. Second example is sickle cell anemia. It's caused by mutation in the HBP gene responsible for producing the beta subunit of hemoglobin. It's inherited from one or both parents. The normal gene is called the hemoglobin A gene. The abnormal sickle cell gene is called the hemoglobin S gene. Huntington's disease. It's a monogenic disease caused by a CAG expansion in the gene coding for Huntington located at short arm of chromosome 4. So the main characteristics of monogenic traits usually exhibit Mendelian inheritance pattern, dominant or recessive. For example, cystic fibrosis is an example of a recessive disease. Sickle cell anemia. People inherited sickle cell anemia disease in an autosomal recessive manner. Huntington disease is a devastating monogenic autosomal dominant disorder. So, what about polygenic traits? Polygenic traits, as I told you before, controlled by multiple genes, often influenced by environmental factors. As the same examples, height, skin color, risk of common disease like hypertension, and so on. So the main characteristics show a continuous range of phenotypes and complex inheritance patterns. And I will explain these parts in the following slides. The genetic architecture of polygenic traits, if we want to explain or discuss this part, we should highlight the additive effect. Each contributing gene, we said that polygenic traits result from many genes, polygenes, multiple genes. So each contributing gene has an additive effect on the traits, the overall phenotype, meaning the total effect is the sum of each gene, each individual effect of each gene. So it's additive effect. Sorry. And there is important term, it's called epistasis or gene-gene interaction. We can define epistasis or refer to a specific type of interaction between genes, where the effect of one gene or allele is modified by one or several other genes. In epistasis, one gene can mask or alter. So there is an interaction, and the interaction may be mask or alter the expression of another gene at a different locus. This means that the genotype at one locus can influence or change the phenotypic expression of the genotype at another locus. Okay, why, someone may ask me, why you mention epistasis, why we are talking about polygenic traits. So the relationship between epistasis and polygenic traits in that, while polygenic traits involve the additive effect of many genes, epistasis describe how these genes might interact in non-additive ways, so potentially altering the phenotypic expression of the trait. We know that genes in the polygenic trait can affect the final phenotype by their additive effects. But in epistasis, describe how these genes might interact in non-additive way, mask or alter, gene-gene interaction. This interaction is a key factor in understanding the genetic architecture of the complex traits. Again, another interaction, gene-environment interaction, occurs when the effect of an individual's genetic makeup on a trait is influenced by environmental factors. In other words, the same genetic variant may lead to different outcomes depending on the environment that's surrounding the individual in his life or her life. So what is the relation between gene environmental interaction and polygenic traits, and how it's related to the genetic architecture of polygenic traits? Let me give you an example. Consider a polygenic trait like educational attainment. And educational attainment, as I told you, is one of the polygenic traits. It's educational attainment is influenced by many genes. Environmental factors such as quality of schooling, parental involvement, and socioeconomic status can interact with genetic predisposition to influence educational outcomes. So, two individuals with the same polygenic risk score for educational attainment might achieve very different levels of education. Why? This is based on the environment they are exposed to. So, to sum up, the interaction between genes and the environment is critical in shaping polygenic traits. Understanding this relationship is essential for predicting outcomes and develop personalized interventions that consider both genetic predisposition and environmental influences. Another point, very important polygenic trait, is quantitative nature. The quantitative nature of polygenic traits refer to the way these traits are influenced by the combined effect of multiple genes, the additive effect of the multiple genes, each contributing a small amount to the overall phenotype. These result in traits that are typically continuous and measurable. They are not discrete. So that's why they are often called quantitative traits and can be represented as a normal distribution, bell shape, as I will show you. So polygenic traits often follow a bell curve, normal distribution population due to the additive effect of multiple genes. Let's show you this here. Okay. This is the normal distribution, bell shape. The graph shows there are many possibilities. The most people have the average or medium. If we consider y axis for frequency of phenotype 10 to 20 and so on, and x axis it is the phenotype in feet. For example, in the between 60 to 80. So the average may be 70. So the average, most people have the average or medium version of the phenotype, while the number of individuals, a small number of individuals at either extreme here, less than 70 or more than 70 on both sides. Okay. This is the quantitative nature of polygenic trait. Another point in the quantitative nature of polygenic traits is heritability. Heritability measured between 0 to 1, and it refers to the proportion of variation in a trait that can be attributed to genetic difference among individuals in a population. For polygenic trait, heritability is typically partial. So if we have polygenic traits, or we actually identified all genes that affect a certain polygenic trait, heritability play partial role in this. And why we say this? Because both genetic and environmental factors contributed to the observed variation. So heritability here typically partial. Okay, and if we wanted to study polygenic trait and know the heritability or measure the heritability, we should use twin or family studies to estimate the heritability. Example of polygenic traits and their research: height. There are many genes involved, like LCORL, BTCH1, GPR 126, and HMGA2. Environmental influence such as nutrition during childhood. Skin color, multiple genes involved, like MC1R, SLC24A5. They are related to pigmentation. For environmental influences like sun exposure and ultraviolet radiation. For common diseases like type 2 diabetes involve numerous genetic variants, TCF7L2, FTO. For cardiovascular diseases, polygenic nature involve multiple risk alleles, LDLR for these genes. LDLR, APOE, very common. When you are studying polygenic traits and make studies, what are the many challenges that you will face? Number one, it's complex. Complexity. Difficulty, I told you from the name polygenic, many genes involved. So difficulty in identifying and studying all contributing genes due to the large number involved. Another point represent a challenge in studying polygenic trait, environmental influences. Separating genetic effect from environmental effects can be challenging. Lifestyle factors like diet and physical activity affecting traits like obesity and cardiovascular risk. And the third one, population differences. Variability in genetic contributions to traits across different populations. So it's important to study, if you want to study a polygenic trait, it's very important to pay attention to the population that you will use in studying this polygenic trait. Importance of studying diverse population to understand the full spectrum of genetic influences is very important. Regarding research techniques for studying polygenic traits, there are two research techniques. Number one, genome-wide association studies. It's used to identify genetic variants associated with traits across the genome by scanning the entire genome. The process involves, you will bring, the study design involve two groups. One with the trait and the other act as a control without the traits. So involve comparing the genome of individuals with and without a particular trait. Okay. Example of this was identifying multiple variants related to polygenic trait like height. Research techniques also, the second one, polygenic risk score, and Dr. David will give you in detail in the next lecture. For polygenic risk score, it represents aggregating the effects of multiple genetic variants to predict an individual's risk of developing a trait. And it's used in the prediction of the risk of complex disease like heart disease, diabetes, and cancer, breast cancer, prostate cancer. For ethical considerations in polygenic trait research, there are four main topics: informed consent, privacy and data protection, potential for discrimination, ethical use of polygenic risk score. Let's start with informed consent. For informed consent, there are two important points. Number one, you should ensure that each participant is fully aware of the information regarding study purpose, procedures, risks, and benefits. So participant autonomy is very important. In addition, transparency. Each participant should be fully aware of how their genetic data will be used, stored, and in case you will do collaboration inside the country or outside, how their genetic data will be shared. Another point in informed consent related to the process. Use layman's terms to explain complex genetic concepts.
In addition, each participant had the right to withdraw from the study at any time without penalty. So it's a voluntary participation. The second point is privacy and data protection. So confidentiality measures very important. This is in the form of anonymization, remove identifying information from genetic data to protect participants' identities, secure storage, implement robust cybersecurity measures to safeguard genetic data against breaches. For privacy and data protection, also, there are two regulatory framework very known, and you should be aware of this if you will do research in USA or in Europe. In Europe, there is GDPR, very important to know this, General Data Protection Regulation. It's a European regulation governing processing of personal data, including genetic data. Um, ensures is very strict data protection and privacy standards. It's adopted by the European Parliament in 2016 and become very effective in 2020. For HIPAA, Health Insurance Portability and Accountability Act, provisions relevant to genetic data protection in USA. Potential for discrimination. How? There is a risk for employment discrimination or insurance discrimination. Employment discrimination concerning about employers using genetic information to make hiring or promotion decisions. For insurance discrimination, risks for insurance companies denying coverage or increasing premiums based on genetic predisposition. As a preventive measures, there is GINA, abbreviation for Genetic Information Non-Discrimination Act in USA. It was released, or the enactment of this act was in 2008 and became effective in 2012. And it protect against genetic discrimination. Also, we are advocacy need for global policies to prevent genetic discrimination.
Ethical use of polygenic risk score, preventive measures for in the healthcare context, yes. For personalized medicine, using BRS to tailor prevention and treatment plans ethically. For patient counseling, ensuring patients understand the implication of their polygenic risk score results. And avoiding misuse. This is very important in the ethical use of polygenic risk score. Non-medical context, preventing the use of PRS for non-medical purposes like predicting educational attainment or athletic ability. Regulatory oversight, need for guidelines and regulations to oversee the applications of polygenic risk score. Regarding the significance of studying polygenic traits, it's mainly related to disease prediction, personalized medicine, and public health. For disease prediction, understanding genetic predisposition can help in predicting the risk of complex diseases. For example, polygenic risk scores for predicting the risk of conditions like coronary artery disease, cancer, in case of cancer, breast cancer, prostate cancer, and others. Significance also for studying polygenic traits, personalized medicine, tailoring medical treatment based on individual genetic profiles, pharmacogenomics to determine the most effective medication based on genetic makeup, public health, developing strategies for disease prevention and health promotion based on the genetic risk profiles. Population screening programs is very important for identifying individuals at high genetic risk for certain disease. It's very important. Okay, let's start with case study in polygenic traits in personalized medicine. What do you mean by personalized medicine? Personalized medicine involves tailoring medical treatment to the individual characteristics of each patient, including their genetic profiles, genetic profile. So, this example include breast cancer risk prediction for patient, a 40-year-old woman with a family history of a breast cancer. Analysis of multiple genetic variants to this patient to calculate polygenic risk score for breast cancer. The patient is found to have a high polygenic risk score, indicating an increased risk of developing breast cancer. So, for preventive measures, these may lead to enhanced screening, measure frequent mammograms and MRI scans. Also, lifestyle modification, more recommendations for diet, exercise, and other lifestyle changes to reduce risk. Prophylactic measures, consideration of preventive mastectomy or chemoprevention. For personalized medicine in case if the cancer is detected, treatment plans are customized based on the patient's genetic profile to improve efficacy and reduce side effects. So it's personalized, tailored to the genetic makeup of the patient. So lesson learned from this case, benefits, challenges, and future direction. For benefits, early detection increases chance of detecting cancer early, leading to better outcomes. From frequent screening, we can detect cancer early and change the lifestyle of the patient and so on. Start targeting intervention, more effective prevention and treatment strategies. For challenges included in this, two main challenges. Access to testing, we should ensure equitable access to genetic testing and personalized medicine for everyone. Interpretation of the results, the presence of genetic counselors is very important. It will help patients to understand and make informed decisions based on their polygenic risk score. So the main challenge here, equitable access and genetic counselor for the interpretation of the results. For future direction of polygenic risk score integration, wider integration of polygenic risk score in clinical practice. And the research is still ongoing to improve the accuracy and utility of polygenic risk score in various populations. What is the applications in African contexts? Genetic diversity. You know that Africa is the most genetically diverse continent, which provides a unique opportunity to study the full spectrum of human genetic variation. This diversity is due to the fact that modern humans originated in Africa and have had the longest time to accumulate genetic mutations. For health implications, understanding polygenic traits in African population can lead to better insights into the genetic basis of the diseases that disproportionately affect Africans. This can improve diagnosis, treatment, and prevention strategies tailored to these populations. Also, most genetic, as you know, as a global health equity, most genetic research has focused on European populations, leading to a major gap in knowledge about other populations, especially African populations. Studying African populations can help bridge this gap and ensure that benefits of genetic research are equitably distributed. For the current research in Africa, the African Genome Variation Project, AGVP, this project aims to create comprehensive catalog of human genetic variation in sub-Saharan Africa. This project involves sequencing the genomes of individuals from different African populations. The main goal to understand genetic bases of complex diseases and traits in African populations, which can lead to improved healthcare outcomes. It has a major impact. Data from AGVB is helping to identify genetic variants associated with diseases like hypertension, diabetes, infectious diseases like malaria and HIV. Second one, the current research, H3 Africa, or Human Hereditary and Health in Africa. It's a consortium that supports population-based studies that use genetic, clinical, and epidemiological data to address health issues in Africa. It includes many projects supports a variety of projects, including studies on the genetic basis of non-communicable disease like heart disease, diabetes, and infectious disease like tuberculosis and HIV. For future direction for Africa, three main issues. Number one, more data collection. Number two, integration with clinical data. Number three, capacity building represented a more training infrastructure and training for data collection, more genomic data from diverse African population. When you say African population, you should say African populations is needed. So it's not only one population, African populations. There is a variability inside Africa. So from diverse African populations is needed to fully understand the genetic architecture of polygenic traits, integration with clinical data, combining genetic data with clinical and environmental data can enhance understanding of gene environment interactions. Also capacity building, investing in infrastructure, training, and resources to build local capacity for genomic research in Africa. Also, more collaboration, encouraging international collaboration to facilitate data sharing and resource pooling. Ethical considerations, addressing ethical issues related to genetic research, including informed consent, privacy, and the potential for stigmatization or discrimination. To sum up our lecture, number one, polygenic traits are influenced by multiple genes and environmental factors. Also, polygenic traits, they exhibit continuous variation and complex inheritance patterns. In addition to understanding polygenic traits is crucial for advanced personalized medicine, disease prediction, and public health strategies. For future direction, continued research on polygenic traits in diverse population is highly recommended and it's very important. In addition to, we should take care of this point, integration of genetic information into healthcare. This will lead to better disease prevention and treatment. Thank you.